is a neurotransmitter that binds onto receptors through synaptic signaling. Since different neurotransmitters are made in different parts of the brain, the type of dopamine receptor that I'll be focusing on is generated in the pars compacta of the substantia nigra, which is part of the midbrain. There are 4 types of dopamine pathways in general: the nigrostriatal pathway, mesolimbic pathway, mesocortical pathway, and tuberoinfundibular pathway. So since today I'll be talking about the dopamine that relates to movement, we'll be looking at the nigrostriatal pathway, meaning it'll control motor function. So if there's a positive influx of dopamine, that could result in chorea, athetosis, and syndromes such as Tourette's and tics. If there's a negative efflux of dopamine, that could mean that it could result in Parkinson's disease or a dose of antipsychotic drug effects, such as cocaine, where everything is slowed down. So first, dopamine that's already been synthesized from the midbrain sits in these vesicles called VMAT2 transporters, and as the nerve signal reaches the axon terminal, it stimulates the calcium ion channel to open and causes an influx of calcium ions into the presynaptic neuron. This causes the VMAT2 vesicles to fuse with the cell and release their neurotransmitters into the synaptic cleft. Now, one of 2 things could happen. The neurotransmitters could go to an autoreceptor, inhibiting the exocytosis of dopamine, and regulate the excitability of the dopamine neurons that the body uptakes, or a negative feedback. Or what we'll be focusing on is binding onto a receptor onto the postsynaptic cell. In the scenario that a neurotransmitter binds to one of the postsynaptic G-coupled proteins, the G-alpha subunit will detach and reattach itself to adenylyl cyclase, which then becomes activated. CAMP is then produced by the cyclase, which then activates protein kinase A. pKa will phosphorylase a protein, which will then express CAV1, the gene for stimulating excitability. pKa can also activate DARP32, which inhibits protein phosphase 1, or PP1, preventing the excitation protein from being dephosphorylated. Dopamine then gets recycled back to the presynaptic neuron through the dopamine active transporter, which resets the entire response as explained. Dopamine can also elicit a negative response when it binds to the D2 receptor protein. The G protein connected to the D2 receptor splits into alpha and beta gamma subunits. The alpha subunit inhibits adenylyl cyclase, which means there's no excitability response, and the beta gamma subunit goes in to inhibit CAV1, which results in a depressive response. After dopamine causes the response here, it can go back into the synaptic cleft, where eventually enzymes will break it down, or it can go back into the presynaptic neuron through an active transporter. Once in the presynaptic neuron, it can be broken down or metabolized by monoamine oxidase, or go back into a VMAT2 vesicle.